Hello guys, welcome back to my channel. This video is about metabolism of xenobiotics. It's a biochemistry topic from MBBS first year, and it's a pretty short and simple topic, and I'm referring to it as metabolism of xenobiotics rather than detoxification. And why, we'll know through the course of the video. And throughout the video I'm covering all the points of the chapter, which is of metabolism of xenobiotics. I'm going to focus more on the concept of the chapter rather than the points that require a lot of memorizing. So you can go through this video, and then at the end of it you can go through the points which require, or the parts of the chapter which require a certain extent memorization. Also, I've referred to Satyanarayan, and so the whole lecture will be word to word from Satyanarayan. So let's get into it. So first of all, whenever we start biochemistry, to make it more relatable to our subject, to medicine, we talk about why it is required, right? Any chapter or anything, why we are discussing about it. Because as humans, we are exposed to, we cannot imagine how many foreign compounds, like we eat so much, there are so many pollutants around, we wear so many things on our bodies, we apply so many cosmetics, creams, soaps, sprays, deodorants, whatever, 1,000, a million stuff is there. So apart from that, those substances can also get into our system, and also apart from that, the bacteria in a large intestine, they convert certain products in our body into some foreign substances. That means substances which are produced by the body but are toxic for the body. So now we have to do, what we have to do is we have to detoxify the body. We have to remove these toxic substances, these foreign substances. For example, indole is formed from tryptophan, which is toxic. Indole is toxic. Lysine, say we get cadaverine, which is toxic. We get tyramine from tyrosine, again toxic. Phenol from phenylalanine. All these are products in our body and they're toxic. And one of the most heard one, and the one which we all know about, is bilirubin. So bilirubin is something we have studied so much about, and it is also something, it's a end product of metabolism, and it has to be excreted out. So even that is an example of a toxic substance that is produced in our body and has to be excreted out. So detoxification or detoxification is a series of biochemical reactions occurring in the body to convert the foreign, often toxic compounds to non-toxic or less toxic and more easily excretable forms. So you're talking about why detoxification is a misnomer, is because sometimes when a body is trying to convert these substances into a more excretable form so that it can be excreted out from the body, instead of detoxifying it, it can end up producing a more toxic compound. Two examples for this are given there. One is, if procarcinogens go through this metabolism, carcinogens are formed, which are arguably, obviously, not arguably, more toxic to the body. Apart from that, another example is conversion of methanol to formaldehyde, which we have seen how it affects our body in alcoholism. So in such cases, we can't justify the word detoxification, because the same metabolism is happening but the products are actually more toxic than the original substance. The only difference is they are more easily excretable. So that is why we come up with the term biotransformation or metabolism of xenobiotics, which is a more correct way to refer to this. So now first thing we're going to talk about is the site of metabolism of xenobiotics. It is obviously mainly in the liver, no doubt about it. When we see the functions of the liver, since the beginning of time we've been reading detoxification. So here is when it comes. So because liver has the enzyme machinery, this detoxification are carried out mainly in the liver. Other organs are also sometimes involved, kidney and other organs, but not much. So the site of metabolism is liver, and how it is excreted is through the kidney, not air through the lungs or feces through the GIT. It is mostly through the kidney it is released. So we see that in a very easy example, bilirubin. The conjugation happens in the liver and then it is excreted through kidney. Mostly, right? Mostly it's excreted through the kidney, in the urine. So we see some amounts in the feces also, very, very little. When it increases, then we can expect there is a problem with the liver or problem with the bilirubin metabolism. So yeah, that is about this site. Now coming to the main part of the chapter, that is the phases of metabolism or the mechanism of detoxification. So the two phases, these two phases can happen either separately, or independently, or they can happen together. So phase 1 involves oxidation, reduction and hydrolysis. Phase 2 involves conjugation reactions. So generally detoxification involves both of the above. For example, oxidation followed by conjugation, or reduction followed by conjugation. but the most frequent is oxidation, followed by conjugation. So now we're going to go into detail about these reactions. So we have oxidation, reduction, hydrolysis in the phase 1 of the metabolism of xenobiotics. So coming to oxidation, as I said, large number of products go through this, and the ones mentioned in the book are alcohols, aldehydes, amines and its derivatives, aromatic hydrocarbons, sulfur compounds and drugs. So alcohols and aldehydes give corresponding carboxylic acids, like methanol will give formic acid, ethanol will give acetic acid, benzaldehyde will give benzoic acid. So that is about alcohols and aldehydes. In amines, if we take an aliphatic amine, then we'll get an aliphatic carboxylic acid, COOH. If we are taking an aromatic amine, then we get an aminophenol. We don't get an acid, we get a phenol if it is an aromatic amine. So RCH2NH2 will give RCOOH, but C6H5NH2 will give C6H4OH and H2. That means para-aminophenol on oxidation. Aromatic hydrocarbons, it is interesting. So the example that is given here is benzene on oxidation will give 4 products. One is a phenol or monohydroxybenzene. Then we have quinol and catechol, which are basically, if I say in a layman's language, dihydroxybenzene, but it should be parahydroxyphenol for quinol. So both of them have 2 OH groups, but the position differs. Here you have it at the para position, here you have it at the position just next to here, which I stupidly have forgotten. Right, so that is quinol and catechol, and then the last one is hydroxyquinol, which is dihydroxyphenol. So if I try to make it, it should be OH here, and OH here. So that is about the aromatic hydrocarbons. Sulfur compounds, organic sulfur is oxidized to sulfuric acid. Done. Now talking about drugs. So there's one example given which is important. So there's a drug called mepropamate, which is a tranquilizer. So it is oxidized to give what? We are obviously giving iron hydroxy groups here. So hydroxymepropamate. Alright, so this is excreted in the urine. So that is basically the only job of this metabolism, to make more easily excretable products. So mepropamate is converted to hydroxymepropamate, which is excreted in the urine. Coming to reduction, same thing, the products are reduced to a more easily excretable form. Examples given are picric acid to picrimic acid, nitrobenzene to aminobenzene. And there's one example of chloral. So chloral can either get oxidized to trichloroacetic acid, or it can get reduced to trichloroethanol. Both trichloroethanol and trichloroacetic are more easily excretable. So here chloral can go through different metabolisms of detoxification. So that is something I've noticed. Coming to hydrolysis. So what are you doing in hydrolysis is breaking up the bonds, ester bonds, glycosidic bonds, amide bonds. So breaking up that is basically how you're doing hydrolysis. So the examples given in the textbook are aspirin, diisopropyl fluorophosphate, atropine and procaine. So at least remembering these examples is important, and then if you can remember the products also, it is even better. So aspirin, which is obviously acetyl salicylic acid, it is actually giving you salicylic acid and acetic acid, which are then excreted. Then diisopropyl fluorophosphate will give a dialkyl phosphate and hydrogen fluoride. So it's basically the fluoro is going away, right? So dialkyl phosphate is being left out. Atropine on hydrolysis will give a tropic acid and tropine. Procaine will give para-aminobenzoic acid and diethyl aminoethanol. So all this is only memory based, so you can do it on your own. So that is about the phase 1. So one thing that I have to mention is about cytochrome P450, which is most of the oxidation reactions of detoxification are catalyzed by cytochrome P450, which is a monooxygenase, or also called as a mixed function oxidase. So why is it called as P450? Because there's an absorption peak when the enzyme is exposed to carbon monoxide. That's why we call it cytochrome P450, peak at 450 nanometers. So where do you find it? You mostly find it in majority in the microsomes of the liver. Very little amount is present in the mitochondria of the adrenal gland also, but mostly microsomes is where you find this enzyme, or this is actually called as the hemoprotein. It has heme as a prosthetic group. So what does it do is that most of the reactions, they involve the addition of a hydroxyl group because oxidation is there. So aliphatic or aromatic compound gets added with the OH group. So here you can see this is the compound. RH is becoming ROH using of oxygen, so monooxygenase, right? Correct. So as we can see in this reaction, one more thing is NADPH is getting converted to NADP plus. That means the mechanism of action of cytochrome B450 is complex and is dependent on NADPH, right? So that is something that you remember. And multiple forms of it are existing. 20 to 200 forms are there, and that is a different case altogether. Then 3 more points about it is there's a phospholipid phosphatidylcholine, which is actually the constituent of the system and is necessary for its action. It's a point given. I currently don't have any explanation for it. And also it's an inducible enzyme, right? In the inducible enzyme, that means it goes back to your translation and those regulatory mechanisms and everything in molecular biology. It's a hemoprotein, as I said. So it's an inducible enzyme because its secretion is increased when on administration of phenobarbital. Apart from cytochrome P450, there's a distinct species, namely cytochrome P448, which has been studied, and it is specific for the metabolism of polycyclic aromatic hydrocarbons. Polycyclic aromatic hydrocarbons. It is also known as the aromatic hydrocarbon hydroxylase, right? Okay, done. So coming to the last part of, actually second last part, phase 2 of the mechanism of detoxification, conjugation. So conjugation is what? You're taking a foreign compound and you're taking a compound from your body, you're conjugating them together, and it's getting excreted out of the body very easily. And we've seen this already in the liver during the metabolism in, I think in cholesterol metabolism, I am not sure. So that bilirubin gets unconjugated bilirubin gets conjugated to give conjugated bilirubin in the presence of glucuronide transferase, and it's then excreted out. It's actually taken to the kidney, and from there it is excreted out. So that is what is happening in the conjugation reaction, and most of the times it is followed by the phase 1 reaction, mostly oxidation. So there are 8 different agents, or we can say 8 different conjugating agents which have been identified in the body. They are glucuronic acid, glycine, glutathione, glutamine, methyl group, sulfate, acetic acid, and thiosulfate. And we're going to quickly discuss all of these. Whatever is there related to concept, I'm going to tell you about that. So glucuronic acid, as I already mentioned, it happens in bilirubin. The active form of glucuronic acid is UDP glucuronic acid. So any compound like bilirubin combines with UDP glucuronic acid to give conjugated bilirubin. That means bilirubin, you can say glucuronide, and you'll get UDP. The enzyme is UDP glucuronide transferase, which I think most of you know. One thing that you might not know is that it is induced by barbiturates. So we saw phenobarbital here, and we are seeing barbiturates here. So barbiturates are when administered, they induce the glucuronide transferase enzyme, which increases this conjugation reaction. Next, coming to glycine. Glycine mostly acts on aromatic carboxylic acids like you could say benzoic acid, or benzoic acid may, if you add a CoA, then it will become benzoyl CoA, or phenylacetic acid, or cholic acid. What happens in the reaction is simple. Glycine gets added, you will get a product. So like phenylacetic acid will give phenylacetic acid, cholic acid will give a glycocholic acid. The one important is that I mentioned here is benzoyl CoA to benzoyl glycine, right? So that is nothing but hippuric acid. So here I'll probably write it's the benzoyl glycine, right? So aromatic carboxylic acids. Next, coming to glutathione, and along with that I'll discuss acetic acid also. So here we are basically seeing how mercapturic acid is formed. Alright, so the tripeptide glutathione, it does not have a separate active form. It itself is an active conjugating agent. And what happens in the reaction is that compounds like aryl or alkyl halides, alkenes, nitro compounds, epoxides, they get conjugated with cysteine. Why cysteine? Because glutathione is a 3 amino acid, you could say tripeptide, right? With glutamate, cysteine, and glycine. So only to the cysteine the substance gets conjugated with. How? Let's find out. So imagine this Rx is the substance and this is glutathione. We see glutathione transferase. So the X is removed and the glutathione gets attached here to give RGS. Now what we are doing is slowly we are removing glutamate first, then we are removing glycine for only cysteine to be left out. So in the first reaction we have alpha glutamyl transpeptidase. That means this glutamate is going from here, it's probably going to some other compound, that's why it is called as transpeptidase. It's going to some other peptide. You are getting R cysteine glycine because glutamate is no longer there. Next, you're removing the cysteine from this by enzyme cysteine glycinase. So the you're removing glycine from it. Cysteine glycinase, so the glycine will go away. What is left out? R cysteine. Excuse me, what is wrong? 1 second. R cysteine. Now the last reaction. So you would say at this step it's already conjugated, right? Because R is there and cysteine is there. But what we are seeing currently is the conjugation to form mercapturic acid. And in that there are two agents that take part. One is glutathione that we already discussed. The second one is acetic acid. So acetic acid, mostly the active form is acetyl-CoA. So one example here is with the sulfur drug. Sulfanilamide will give acetyl sulfanilamide. This is the conjugated product. This is excreted out. Even here, R-cysteine gets conjugated with acetyl-CoA. The enzyme is N-acetyltransferase and the product is mercapturic acid. So to form mercapturic acid, basically conjugating a certain product to give mercapturic acid, there are two agents that are involved, glutathione and acetic acid. Glutathione is itself the active compound, but acetic acid ka active form is acetyl-CoA. Next, coming to glutamine, it's relatively less important. Phenylacetic acid gives phenylacetylglutamine. Done. Methyl group and sulfate may again we'll see a separate active form, right? In methyl group we have S-adenosyl methionine. In sulfate we have 3-phosphoadenosine 5-phosphate or PAPS. Both of these case, in this case the methyl is added here, so when methionine se methyl group is gone, it forms homocysteine, so S-adenosyl homocysteine and your conjugated product. Here you'll have your phenyl sulfate from the PAP, and then the leftover, the PAPs becomes PAP basically. So these two are the conjugated products. Here the enzyme is methyltransferase, here the enzyme is sulfotransferase. Done. The last is thiosulfate. So next, coming to thiosulfate. So in thiosulfate, the reaction you simply remember a cyanide plus sodium thiosulfate will give sodium sulfate and thiocyanate. Cyanate. So cyanide is getting converted to cyanate, right? You see this here. So coming to thiosulfate, in here the highly toxic cyanide is getting converted to less toxic thiocyanate. So the reaction goes like, you have cyanide and you have sodium thiosulfate, which is giving you thiocyanate, which is less toxic, and whatever is left over is sodium sulfate. So that is about the all 8 conjugating agents which are responsible for the conjugation reactions in our body. So ye to 2 phases are done of detoxification. The chapter is over. But there's one more thing that is mentioned, that is detoxification by drugs. Now why is this separate? Because the mechanism by which these work are not known. In just now the phases we discussed, we know exactly how the detoxification is taking place, what are the steps: oxidation, reduction, hydrolysis, conjugation, blah blah blah. Here we don't know the mechanism, we just know that it works. It combines with metals and it gets excreted easily. So the example is British antilvice. I do not know how to antilvisate. I don't know how to pronounce it properly. British antilvisate. BAL is what I'm going to call it. So that overcomes the toxic effects of certain metals like arsenic, mercury and cadmium. This was used during World War II as a detoxifying agent for certain war poisons. The mechanism is not known, just that combines with the metals, gets excreted easily, does the detoxification. So that's all. It's important to mention that it acts like an applied aspect of this topic, right, of the process of detoxification. So yeah, that's all for this chapter. Just go through the reactions once again, so because you have to remember them. I'll try my best to make it easy for you. Thank you so much for watching.